Thank you very much for the invitation to speak. And I'm going to try and explain some of the things that we are doing in collaboration with many people. I'm going to introduce some of them at the end of the talk. And basically the idea here is we are trying to use mathematical tools, or the mathematical tools that we are familiar with, namely topology, in trying to understand something about the brain. Okay, so let's start thinking about what the brain is. So we are more or less convinced that we all have one. You know, we can do various things with it. And in fact, we also know that there are lots of other organisms, in fact, almost any organism that is more than a bacterium has one. And so, for instance, the smallest brain that I think science is aware of belongs to this little thing called rotifer. And it's really a very, very tiny brain, but still a brain. You have to have a microscope to see the organism itself, nonetheless, its brain. The fly has a brain, which is, I'm going to talk about a little bit later on, not in a very positive way. And we also think that us as humans have the largest, most developed brain, but actually developed, I'm not sure, but largest is simply not true, because here are two bigger brains, bigger in terms of the number of cells in it, of the African elephant and the long-fin whale. That I think we are pretty lucky that they don't have hands and ways of speaking to each other like a language like ours, or otherwise we'd probably be overrun by them. But anyhow, so let's think about what is the brain and what is it good for? Okay, so what do we do with the brain? If you ask a person, what do you do with the brain? Then the normal answer would be, well, you think with it, which is true, but that's not all you do with the brain. I mean, there's a lot more that you could do, or the brain does, whether you like it or not. For instance, the brain commands any voluntary movement that we have. Okay, it also controls most internal body functions, from heart rate and all the rest of it, you know, the digestive system and so on and so forth. It's responsible, in fact, to at least in part for every sense, feeling, emotion, and whatever you can think of, the brain is controlling in one way or another. But the question is, and the mystery is, how does it do any of those things? Okay, how does it do it? So, well, how can we learn the answer to this question? Well, one way is to try what we do in science fiction. You know, you put a bunch of electrodes on top of the brain and you connect it to some big machine. And like in the movies, you are able to read the mind of the person that is being examined. But that, of course, is still very much science fiction. More seriously, we have the EEG, the electroencephalogram, that is a medical device, and functional MRI, from which you can get a lot of information about higher level activity in the brain. So these things could give you information, for instance, answer the question like, what is the brain doing in the sense of, is it active in a certain part of it? Does it react to a certain stimulus that's coming from external senses? Which parts are active? What is the response to certain drugs, etc., etc. But it cannot actually, these tools cannot explain to you how the brain is doing it or exactly what the details of the response are. For instance, you can learn that the brain is responding to, like the brain of a mouse is responding to seeing a cat in a certain way, but it cannot respond, it cannot tell you whether the cat is yellow or black. Okay, so to understand the lower level activity of the brain, you need to go into the lower level structure of it, and so to understand how the brain is built. And the brain, or every brain, from the rotifer to the African elephant or the human, all brains are built of cells called neurons, which is probably something you already know. Here's a picture of one. These are tree-like objects with a cell body right here, and they connect to each other. what's a brain and what it's good for. We know that we have one. I said that already. We are not world champions. The long-filled whale and the African elephant have bigger ones. And of course, these two little things, this is the rotifer, microscopic organism, and the housefly, they also have brains much smaller, but the housefly is complicated enough actually to cause us a lot of trouble. And I'm going to talk about this a little later. So what's the brain? What it's good for? We know we think with it. We know it commands voluntary movements. We know furthermore that it controls most internal body functions and responsible for every sense, feeling, emotion, thought, whatever. Okay, all that I already said. Yeah, right. So how does it do all that? And the honest answer is that nobody knows. Okay, nobody knows how it does it. So how can we learn what the brain is actually doing and how it does it? And what I said is that you could go like in the movies, connect some electrodes, fancy machinery, and then you see what the person is thinking, feeling, seeing, whatever. This is all science fiction. This is not how things work, unfortunately, not yet. Okay. We do have some machinery which is similar to what you see here on the left. On the right is a picture of a person with an electroencephalogram, EEG machine connected to himself. And it is also the functional MRI that you can get a lot of information from of higher level activities. But to learn about how the brain actually works on a lower level, on the detailed level, we need to understand what it's built from. Okay, so we need to go to the cell level. All right, so the brain is made of neurons, and here's a picture of a neuron with a cell body, the soma in here, and it has all these branches that are used to connect to other neurons. Okay, so it's not moving yet. Whoops, moved too fast. Right. So when you put some neurons together, then you get a network of neurons connected to each other. And the way in which they are connected is not just touching each other. They actually have a very interesting device that enables them to connect, and that device is called the synapse. Okay. So I'm going to show you a picture of a synapse. So here you are, two neurons. Here's one and here's the other. And the connection between them is a microscopic connection. Mind you, a neuron is about 2 microns, so 2/1000th of a millimeter in size. It's tiny. The synapses are, of course, much smaller, and they are chemical connections. That's what's important to know about them. They're chemical rather than electrical connections. They transmit electrical current, but they're chemical connections. So you have to actually break a certain barrier in order to transmit from one neuron to another. That makes them very flexible and very efficient in doing certain things. But on the other hand, it makes the connections very unreliable. So sometimes a neuron that transmits to another neuron gets the signal through, and sometimes it doesn't. And it's very, very difficult to predict whether a synapse will respond to a signal or not respond to a signal. That makes it the task of understanding what the brain is doing, of course, much harder. We say the brain activity in the brain is noisy, exactly because of that feature of things. Okay, so what do you do to study the brain? Well, one thing you could do is to probe into a real tissue in a wet lab. You need to commit murder for that, because you cannot look at tissue without actually killing the owner of the tissue. So this is done a lot in labs, and we learn a lot about the brain from doing things like that. You can just use the tissue for a while; it's alive for a while. You can also probe electrodes into the brain of a living creature. Again, this is done on lab animals. But the means, it's very difficult to do that, especially because of the size of the cells you're looking at. So because of that, the Blue Brain Project, which is our main collaborator in this project, took the task upon themselves to reconstruct brain tissue on a supercomputer. And what you see here is basically some pictures. This is not what things actually look like, but if they could show you in a picture what it is that they construct, those sort of pictures will be what it would look like. You see here a bit of brain tissue. Here you see a bit of the cortex. So many objects like this, like columns like this put together and making up a cortex, the external part of the brain. And here you see an enlargement of what it would look like if you could look through a microscope in what it is that they're doing. This project actually has produced some amazing results, which are completely neuroscientific, nothing to do with what we're trying to do mathematically. And in particular, this is not just a bunch of nice pictures. It is a model that you can study from every possible aspect of neuroscience. You can study the structure of the brain. You can study also how it responds to certain stimuli. You can stimulate the brain, and you can this artificial brain, and you can look at the response of the tissue and see how what happens. This is not to say that you can read off what happened, but you can see that it's responding, and then you have all the information you need, which is very much unlike what happens with real tissue. You can, from a real tissue, you can get maybe, if you have state-of-the-art equipment, you can maybe read off maybe 100 neurons or a few hundred neurons, no more than that. With this, you can get the response of millions of neurons. Okay, so in some sense, if you get it right, then you have really something that is very efficient, a very efficient machine with which you could try to study the brain. Okay, so we're very lucky to have them as collaborators. And so here's an example of what what happens when the brain tissue gets a stimulus. So here's a little movie for you. And you see here, I hope this works well on the other side, the way you see it, what happens when a signal comes in and the neuron starts responding. Okay, so you see all these flashes that you see there is neurons firing. They are receiving electrical current from somewhere, like at the bottom of the bottom layer here, it's receiving some electrical current, and then the neurons are communicating with each other and respond to that electrical current. So here it is again. You can see these flashes, and then the signal comes in and the whole system goes on fire. Okay, for a while, everything is happening, and then it has to rest for a little while. So that's all very nice and very impressive. This is very good for making movies like that is very good for getting funding. You need tens, maybe hundreds of millions of pounds to create a project like that. So this is very impressive, but you don't learn much from it, because this doesn't tell you anything that you want to know, or what the brain is actually doing when you see this movie. So to try to understand what the brain is doing, you need to look at the response of the neurons. So what are the neurons? When are the neurons firing? Okay, so one way in which you can decode and encode this is by what is called a raster plot. So what you see here is an example of a raster plot, and basically every horizontal line on this plot corresponds to a single neuron. Every dot corresponds to an instant when this neuron actually emitted a spike, when the neuron fired. And so when you record all of them together and you record the firing, you get some sort of a pattern that tells you something about the way in which the system responds to the stimulus. Okay. Problem with that is that it really doesn't matter in which order you put the neurons. Okay. So if you change the order, then they'll get a different picture, a slightly different picture. So these raster plots can tell you how much activity there is. It can tell you that there is an activity, but it cannot really tell you much beyond that. So it's still a very, very crude measure of what's going on when you put a stimulus in. But it's a very common tool for neuroscientists anyway. So what does, so far for crash course in neuroscience, I'm not a neuroscientist, so I should really not give any crash courses, but this is the basic knowledge that you need to understand what it is that we are doing. So the next question is, what does mathematics have to do with all of that? So when a mathematician, especially a topologist, sees a brain, reads about the brain, then the thing we see actually is not a brain, a tissue, but rather a graph. Okay, we can think about the brain as a very large graph. I'm going to get to that in a minute, where the nodes in the graph. So this is an example of a graph. The nodes in the graph are the neurons, and the connections between them are those edges, those lines that connect the nodes. Okay, better yet, because neurons are directed, so they have inputs and outputs. They're not just connections, they have inputs and outputs, much like a diode in electronics. Then we see a directed graph, okay, a graph that has direction to it, like this one. Okay, so this is very nice, and you could actually think about, if you want to think about the brain as a graph, it's a very common thing. It's done by scientists for generations now, but there's one little problem, and that is that graph is typically a lot larger than what you're seeing here, a lot larger and much more complicated. So here's an example of a picture of the brain of the housefly. Okay, if you want to see the brain of a housefly as a graph, then that's what it's going to look like. And this is just a part of the brain. So again, not very useful. So just drawing pictures is not very useful. You need some finer tools to deal with things. In fact, I'm going to show you another picture. One of my postdocs a couple of years ago looked at the brain of a worm called C. elegans. C. elegans is a tiny little worm, 0.5 millimeter in size, has 279 neurons in its brain. So it's a very small brain compared to a human brain, which is 100 billion neurons, or even to a mouse brain, which is 10 million neurons. So tiny brain, and he tried to use some software to draw a graph of the brain of that organism. So here it is. Again, not very useful. You can't use pictures. I mean, it's very impressive that it's so dense and so black, but you can't really tell anything about the structure or what it does when it does something. All right. So we need some finer tools. All right. So one of the tools that has been used for many years in neuroscience is graph theory. So people come up with, if you have a graph, they come up with various invariants, various numerical functions that you can associate with the graph that give you a lot of information about features of the graph, general features of the graph, certain nodes, certain clusters, and so on and so forth. I don't want to talk too much about this, so I just put a page of formulas for you. You don't have to remember or even understand any of that, but just to say there has been a lot of mathematics that is used to try and understand the structure of brain graphs that people have been using for at least 20 or 30 years. We are topologists, we are not graph theorists, so we want to do something a little, we are using these tools, but we also have some tools of our own, which we like very much. And so one aspect of using just the graphs is that they're one-dimensional. But when I say one-dimensional, what I mean to say is that they're made of dots and lines, okay? Dots and lines. But look at this picture up here. You see dots and lines as well. These are also graphs. But when you look at this, you say, oh, this is a square, and that's a cube. And if you have your imagination with you, then this is actually a four-dimensional cube. You see, it's a cube that is moving in time, okay? Moving across an interval. So that's a four-dimensional cube. And you can imagine how you can actually generalize this even further and get structures which are even higher dimensional. And similarly, you can look at an object which is very, very useful in topology, which is my specialty. And these are called simplices. These are basically graphs. Again, these are graphs that made of nodes, right? And so here's one with one node, and two nodes connected by an edge, three nodes which are connected by edges, and then you have you fill them in, and then you have the three simplex or the tetrahedron, four nodes connected by edges, and then faces, and then there's the filling in. So this is a three-dimensional object, and again you can go higher and higher in dimension. Okay, so these are some of our tools. And what can you do with that? Now, if you have a graph, you can look at the graph, and whenever you have those cliques, those fully connected triples like this, you fill in a triangle. If you have a fully connected three simplex, then you fill in the three simplex. So you have a geometric object which is made of dots, lines, triangles, tetrahedra, and you can go higher and higher in dimension. This has a name, it's called a Flood complex, a directed Flood complex, but you don't really have to remember it. It's just a geometric object. And we are very proud of these geometric objects. We like them. And when you look at the brain graph and you do exactly that, then you get really high dimensional things. I mean, we got in the blue brain, we got 7, 8, 9 dimensional objects, geometric object. And in one of the models, in fact, the model of the mouse brain, we got a 23-dimensional object. Okay, so it's an amazing structure that you could try to use using the tools of topology to understand what's going on, both in terms of what the structure is like and what the function of the brain is. And when we have geometric objects, then we have tools that we like to use to deal with them. Okay, so I'm going to just mention two of them. One is called Homology, which is a device that allows us to count, in some sense, count n-dimensional cavities or holes in a geometric object. Okay, that's it's a very crude explanation of what it is, and but that's basically what it does. And the number of n-dimensional cavities in a geometric object we refer to as the nth Betti number, after the person that first dealt with these things. Another topological tool is the so-called Euler Characteristic, which is due to the amazing mathematician Leonhard Euler from the 18th century. And that is basically, it's very related to homology, but not necessarily. You can get in two different ways. And it's basically just the sum of the 0th Betti number minus the first one plus the second one, etc. And so that's another invariant of a topological object that you can use, and we use it quite a lot. And I want to give you some examples of what these things look like in what we had in the examples we had before. So here, for instance, you have a connected object. So it's all, everything is connected to everything. So the Betti 0 is 1. And if you count how many one-dimensional holes, so here's a one-dimensional hole, 1, 2, 3, 4, 5, 6, 7. Betti 1 is 7. And there are no two-dimensional holes, and therefore Betti, the higher Betti numbers are all 0. The Euler characteristic is 1 minus 7, which is minus 6. Okay. You want to see an example with a two-dimensional hole. So let's take a look at this object here, where you have all the faces, but it's hollow. Okay, so it's connected. Betti 0 is 1. There's no one-dimensional hole there. You see, there's no holes in this thing, but there's a two-dimensional hole because there's a cavity. And then Betti 2 is 1, and the Euler characteristic is 1 minus 0 plus 1, which is 2. Okay, so here's an example of how we compute these things. Of course, when you have a big object like the graph of a brain and you construct this Flood complex, you don't just do this by hand or by inspection. You have a computer to do it for you, and there are formulas that allow you to do it. Okay. All right, these are the tools. Now let me try to touch on what it is that we do. So let's take a sample graph. Okay, let's take a sample graph. This is not the brain of anything, but it doesn't matter. I don't need to explain what I'm doing. I don't need to explain to you, to give you an example of a real brain graph. It's just a graph. Now assume for a moment that I know, for some reason or other, that some of the nodes in this graph are particularly important. Okay, so here I choose some nodes, one for every color. The colors don't mean anything, by the way. So green, purple, blue, and orange, I chose those. And because they're important, I want to also consider their friends. Okay, so here are the friends. What do I mean by friends? What I mean is exactly those nodes that are connected directly to those central ones. Okay, the ones I chose to begin with. All right. So I chose some nodes and their neighborhood, their friends. Okay, and once I do that, I can also consider, if I want to create a geometric object, I can also consider the connections between them, the connections between all of them. So what we call this? We call this the neighborhoods. Okay, so the red lines are basically all the connections among all the nodes that I have chosen. So the centers and their neighbors, and the neighborhood, or what we call the neighborhood graph of each one of these central ones. Okay. All right. So what is this good for? Well, think about it for a second. Suppose that I could actually see these subgraphs, these red things that I drew here, active in some sense in a brain, okay? You can think about these graphs as symbols or as letters or even as words, okay? And so I could try to imagine a language that is built out of these letters, okay? So for instance, I want to say, let's just imagine that I could look at this and I look at this, the combination of these two subgraphs, and this corresponds to the word I. And the combination of these two subgraphs corresponds to the word love. And those three, when they are lit up in the brain, this corresponds to mathematics. Okay? So if I see those structures, then I could imagine seeing I love mathematics in the brain. All right? Now, this sounds completely ridiculous, maybe because we are not used to it, but think about it for a second. Imagine that you do not know how to read Latin letters, and in particular, you don't understand English. Then this bunch of symbols, why is this bunch of symbols better than this bunch of symbols to say, I love mathematics? All right, so you can create a language, a written language for any symbols, as long as you know what they mean. Okay, the meaning is what's important and not how I write it. All right, so that sounds interesting. Maybe this is what the brain is doing in this. Maybe this is what we are actually doing, but actually, no, I'm lying to you a bit. That's not what we're doing. The reason it's not what we're doing is, well, first of all, the brain is much more complicated than these little graphs that I showed you. And you can't really look at things. You have to associate some of these algebraic, some of these topological invariants to measure, you know, features of those subgraphs. Secondly, as I told you before, the system is very noisy. So you need to not be rigid and say, oh, that means I, that means love, and that means mathematics. You need to be able to somehow operate within some range of uncertainty. So let me show you what it is that we are actually doing. Okay, so here's something coming out of Blue Brain. So this is these are 8 different signals that are coming out of you know, you just insert into the artificial brain, and these are the signals coming in. There come 8 of them, eight different ones. And we have samples of that coming in some random sequence, and we then measure the responses. Okay, when I say random sequences, you see like 0, 5. They have named between 0 and 7, 0, 5, 6, 4, 2, 2, and they just come in random sequence at a random sequence, and then the response. And we record all the responses in a raster plot, like I showed you before, and then we basically select those important nodes in our brain graph. We select these important nodes. We consider the neighborhoods, just like I showed you before. Okay, and for each one of the stimuli, we record the activity and we compute graph and topological invariants of those neighborhoods and create what we call a feature vector. And then we use some standard artificial intelligence, also known as machine learning algorithms, to learn the feature vector of our stimuli and predict the new ones. Okay, so predict an unknown sequence of the same top type of stimuli. So we can say, like, you know, this one is 0, this one is 1, this one is 7, this one is 5, etc., without having seen them before. Okay, so in some sense, we are reading off what the brain, what the artificial brain is doing, and with some certainty. What certainty? Well, we managed to predict the sequence at an accuracy of up to 88%, which is great, because if you're thinking about you know, 8 stimuli that are coming in, very noisy stimuli, the probability of guessing the correct one is 12.5%. Okay, because we have 8 of them. Here we are able to predict them at the accuracy of 88%, which is of course much higher. So we consider this quite a success, especially given the nature of this stimuli. It's very abstract. It's not something specific. It's a very kind of low-level activity in the neural system. And so that's the story. That's part of the story. Of course, there are a lot of other things that we are doing in this group. And I want to just end by introducing the group to you. These guys worked with me until last year in Aberdeen. This is Pedro, my student, and then Dayan and Yanis and Henry and Jason. And these are some of our collaborators in EPFL. Henry Markram, the lead of the Blue Brain project, Catherine Hess and Michael Ryman. And that's about all I have to say. So thank you. Thanks very much for your attention.
Well, thank you very much. I thought that was incredibly interesting. And yeah, it's really opened my eyes to, how you can use topology and neuroscience at the same time. So if you've got any questions, this would be the time to kind of pop them into the chat, but maybe I can start us off while people are thinking of questions. So Ran, how did you get into topology to begin with?
Into topology?
Yeah.
Do you really want to ask this question? Well, I went to university to study computer science because it was supposed to be useful. I did not like it, but I was lucky enough to do this in the Hebrew University, where in computer science you are required to take the first year mathematics for the mathematicians, and that was very challenging, and I loved it. So, I just finished my degree and got a topology course. I thought this was a very interesting thing, and then got into it. But my background is pure topology. So for the first 20 years of my career, I basically did the things that I did not talk about today. And the thing that most of my colleagues in Aberdeen, all of my colleagues in Aberdeen are doing. I got into neuroscience talking to Henry Markram, who's a good friend of mine, about life in general and neuroscience in particular.
So my next question was going to be, how did, what inspired the combination of topology and neuroscience? But you've just answered that. So, clearly just meeting, other colleagues and then end up working with and end up having good partnerships, because clearly this is you're making quite a few breakthroughs, I'm imagining, or getting closer to a big.
So I think there's more to that than just the friendship. I mean, the point is that when you have something as a model as large as the Blue Brain, with so much detail, right? It invites topology. It invites serious graph theory, because you have this huge structure that you have all the details you can hopefully hope for that can be read out. And so once you see that you can just get all the information you need, you say, okay, well, then I can do topology with this. If you take a very tiny, small graph, then there's not much topology there. But once you have, something as big as that, then of course there's a lot of structure, and you can try to study it and see if it tells you anything. So it's exactly the fact that you have this model that allows you to do things in the way that we're doing them.
So are there any animal brains that you would particularly like to investigate and study that maybe you haven't had a chance to? I know whether it's like an endangered animal or, I don't know, just throwing it out there.
Well, so Blue Brain has, the models are rat, mouse, and they aim to do human, but of course human is much more difficult, not because it's bigger, but because you can't kill humans and study their brains. So you have to use the brains of dead humans, and the cells that are dead, and you can't use them in the same way. So there's a lot less information known about human brain than there is about rat brain or mouse brain. I think that if we understood even part of how a rat brain is functioning, that will be a huge breakthrough. So in spite of having all this machinery, in spite of having all this knowledge, the cellular level, genetic level, electrical level, we have all this knowledge, but we have no clue how things are actually happening. So trying to kind of decipher messages, as abstract as they may be, is a great thing, but it's only the first step in trying to understand. So I'm not endangered. I mean, I wouldn't touch endangered animals. You have to kill them first.
Okay, well then that's probably just as well. So are there any questions from anyone watching? If not, I think that's bringing us to the end of this this talk. But I want to. Oh, excellent.
Yes. So I understand that these topological tools are sort of a way to measure complexity of things. So is that the question you're interested in, understanding the complexity of the brain, or what are sort of the questions that you'd like to answer about brains?
So we have a paper that was published in 2017, where one of the things that we looked at is complexity. But that is, I mean, what we were able to show, for instance, is that the complexity of the brain is much larger than a network of a similar size and similar density. But that is interesting, but that's not what I'm personally after. I mean, what I would like to understand more is how can you understand what it is a brain is doing? So you have this big network, and it's doing something. What you see when you look at it is that the neurons are firing, and and then you can measure voltages and things like that. But basically, just neurons firing. And so, how does that translate to anything at all? Nonetheless, all those things that we are aware that we are doing, or any animal is doing. That's really what I'm looking for. Now, how do you do that? Well, that's a good question. First of all, what do you look at? You can't look at everything because it's too big. You can't look at too detailed at every particular neurons, because again, there are too many of them, and it's too small a unit to be functioning on its own. So it's really trying to understand some sort of a coding mechanism using dimensionality reduction that is afforded by the topological tools. So I'm talking to a mathematician, and Marcus is my colleague, so I'm just using language that maybe I cannot, that is not understandable to everybody. But that's basically the answer.
Thanks.
That was a great question and a great answer. So yeah, thank you both. And yeah, I think we might just wrap it up there unless there's any more questions. Well, I'll say on behalf of everyone watching, again, thank you so much, Professor Levi. And yeah, I will just like to tell everyone about our next STEM exchange. So it is going to be delivered by Florence from the European Marine Energy Centre, and we will have a look at different test sites, looking at, in particular, wave and tidal energy and how they can be used and how we can get the most out of them. I don't know that we have a date yet for that, but please just keep checking our social media, and hopefully we'll see a lot of you there for that. Anyway, apart from that, thank you very much, everyone, and well, thanks to Maths Week Scotland as well.